So good evening all of you and welcome back to Excelsior Academy in the series of Leninger Biochemistry. Now we are going to talk about the third part of biomolecules, amino acids. So today we are going to talk about the peptides and proteins. So till before chapters we talked about amino acids, their structures, their properties, the amino acid titration curves, how to calculate PI, how to get the information of pKa, right? So all these were studied. Now let's move on to the peptides. So we know that from amino acids you get the peptides and from peptides you get the proteins. Okay. So first is they are the polymers of amino acids. So they range from small to very large, consist of 2 to 3,000 of linked amino acids. So from this, now I remember, like in the previous class, towards the end, I have asked one question. Anyone remember? I don't remember the values, but I remember giving the assignment. So molecular weight was given, correct? It was 110 Dalton. And then some protein molecular weight was given. So what is the formula to calculate the number of amino acids? How many of you got the answer? Let us say
Ma'am, I got
40,000, let us say. Yes. So what answer you got?
0.00428. I got, ma'am.
No. You can't get that much less. So what you have to do is, it was very, very simple. It was very simple. See, molecular weight of one amino acid is 110, correct? And the protein weight is around 40,000, let us say. Now what you have to do is calculate the number of amino acids. So how many amino acids are present? So what you have to do is, just divide. So 40,000 divided by 110. That's it. So some value you will get. So those are take the round figure. It won't be exact. It will be some 300 or something. I don't know. Okay. So
For
that is,
yesterday's
yesterday.
256.
Yesterday's this value was different. I don't remember. That's why I took
Yes,
40.
ma'am. Yes. For that, I got 0.004.
No, but you cannot get in 0 point. That's what I'm saying.
Okay.
That's what I'm saying. You cannot get in 0 point. You cannot have half the amino acids. Correct. It has to be around. I don't remember. Even if it is 25,000, if you divide by 110, see, around 200 or something it will come. It won't come in 0.5. Okay. So that's what I'm saying. So I'll take more questions, then it will be more clear. Okay. Anyways, so two amino acids molecules, it can be covalently joined through a substituted amide linkage, which is termed as peptide bond, to give you a dipeptide. Okay. So from here, please keep in your mind, there is amino acid 1 and amino acid 2. So you have got one peptide bond. Okay. But it is called dipeptide. Getting the point? Now, when I say tripeptide, it means what? 1, 2, and 3. So it has got two peptide bonds actually. But the entire residue is called tri. Are you getting? So do not get confused, because many times when the numericals are given, they say that there are pentapeptide molecule is given and then calculate this and that. So usually people, penta means what? What they will do, they will take 5 into and they do the rest of the calculation. But it's not like that. 5 is the number of amino acids. But how many peptide bonds are there? 4. Is it clear? Pallavi, Akshita, is it clear?
Yes, ma'am. Yes.
So this you have to keep in mind, okay? So now formation of a peptide bond by condensation method. So what do you mean by condensation? It is nothing but removal of water. So it is nothing but removal of water. Now how exactly it is happening? So see the chemistry behind this, okay? So the amino group of one amino acid, that is R2, let us say this one, okay, is acting as a nucleophile to displace the hydroxyl group of another one. So forming a peptide bond. So how this entire OH plus H, that is water is removed. So condensation means removal of water. Amide groups are good nucleophiles, but the hydroxyl group is poor leaving group. So what is happening? So the amide group, the hydrogen from the amino group is attacking on the OH and hence water is removed. So now what you have to remember is it is C double bond O N H. This is your peptide bond. Okay, it's very simple. Okay, now who can tell me the name of this residue? So what has been shaded in yellow is your peptide bond. Okay, now you have to, if you remember the structure of the amino acids, then you can tell me the name of the residue. So who can tell me, or otherwise I'll pick the name? Yes. Ramya, would you like to try? Which is the first amino acid over here? Yes. Bhavika? See, I told you, that's why you need to know the structure.
Glyceraldehyde.
Glyceraldehyde. No, it is not an amino acid.
Okay, sorry.
Glyceraldehyde is what? Aldehyde, right?
Yes.
Ranjan, do you want to try? What is the name of this residue? So how many
Threonine.
Threonine? No, it's not threonine. First one. This is your first, second, third, and fourth peptide. It means it's a pentapeptide. Okay. Now, what is the first amino acid? Which group is there? It is the OH group. It is the hydroxyl group. So, in the OH group, apart from threonine, what is the another amino acid? Similar to threonine, which is the another amino acid? It is serine. So how you are going to write? It is written as seryl. Okay. Now, second amino acid. Yes, anyone?
Tyrosine, ma'am.
Second is this one. Now tell me.
Glycine, ma'am.
Very good. So it will be written as glycyl. Okay. Now third is this one. This is your
tyrosine, ma'am.
Very good. I hope notes are not kept in front of you. Tyrosyl, yes. Then next.
Alanine.
Very good. Then last. It is
Leucine.
Leucine. Good. So did you see how you have to name the pentapeptide? So first, whatever the amino acids are there, this ill thing you have to do. That is like, you know, the chemistry nomenclature thing. In the same way, like for the alcohol, we always put the OL, correct? So whenever OH is there, we put OL. If it is CHO, we put aldehyde, correct? In the same way, in the case of amino acids, what you have to do is, if it is serine, seryl, glycyl, tyrosyl, aniline, leucine. The leucine will remain as it is. So is it clear? If suppose someone is asking the name of the residue, now can you do it? So even if this is given, suppose this is given with the name, let us say it is given as, let us say that it is given as alanyl methionine. So what you have to do? It means it's a dipeptide. So how many peptide bonds? One. So draw the structure of alanine over here with the peptide bond. Okay. And then make the structure of your methionine. Clear? Okay. Yes. Now molecular weight calculation. So this is what I really wanted to take. This is very important and very easy also. Now, if possible, take out your calculators. Yeah, of course, but in exam it is not allowed. Right now, you can just take out your calculators. Even I'm taking out mine. So that we don't waste our time. Okay. So please remember, as I said before also, so in the book, if you see, the molecular weight of each amino acid is different. Of course, if I take COOH, H, NH2. So this is our glycine, correct? Now, if I take the molecular weight over here, 1, 14 plus 2, 12, again, all these things, if I'm taking, the molecular weight will come around 98 or 95. I don't remember, but something, it is less than 100, okay? But if I have different R group, the molecular weight is going to vary. So what they have done is, in order to calculate, like quantitatively, in order to calculate the molecular weight of protein, what they have done is, they have taken the average of all the 20 amino acids, which was 110 Dalton. Okay. Now, unless until it is mentioned, you have to take by default 110 Dalton. In few cases, if the examiner wants to give, so they will write as 1 amino acid equal to 100 Dalton, or somewhere it is 120 Dalton. Okay. This will be mentioned, but if it is not mentioned anything, then by default you have to take 110 Dalton. Now let's do the calculation. So for example, as I said, 1 amino acid is 110 Dalton. Okay. So hemoglobin, for example, hemoglobin, if I say that it is 48,500 kilodalton. So what will be the number of amino acids over here? Yes, calculate. So I'm ready with my answer. What is your answer?
485 0.
How many zero?
4 zeros. Without calculator, ma'am. Once check.
So this is the answer you're telling me?
Yes, ma'am.
No, it's wrong. See,
440, ma'am.
if it was given 100, Pallavi, okay, then you would have taken 485. Okay? But it is given as 110. So this 10 is going to matter a lot. Someone said 440 something.
Okay, ma'am.
440.
40.9.
0.9. Wrong.
Okay.
You're missing one small but the most important thing. Ranjan, do you want to pin out what they are missing? These values are correct, but something else is also missing. Now, when I tell you the answer, you'll be like, oh, this so we didn't see.
44.9, ma'am.
No,
Yes,
Ranjan,
ma'am.
means you are also missing. Now, I'll tell you what you're missing. Okay, someone has given the answer. Yes, Ramya, kilo. Very good. This
1000 we have to into.
Yes, correct. So this is the point I was telling you that you're missing this point.
I did, ma'am.
So when you're doing 485, 2 zeros are here. What do you have to do? 10 raised to or 1000 you have to multiply. Then
Yes.
divide by 110.
By 110.
4.5. 4.4.
4.4 again? No. So what you will get? So I'm asking number of amino
440.
acids. So you will have
0.44.
0.44. Why 0.4?
449.
This is the value you get. So these are the numbers of amino acid present in 485 kilodalton of protein. I don't know how you're getting this decimal. Decimal you can never get. as I mentioned before also, amino acids cannot be half. At least one should be there, correct?
Yes.
You cannot have in decimals. Decimal, see, if you have something over here, that is because of, you know, the multiplication thing. So whatever in the given option, just see which is in year one. So you will get the answer. Okay. Now, so this was all about the one type of calculation. Now let's move on to the next tricky one. Okay. Don't make silly mistakes. You people are going to write the exam also. Huh. So next question is, so you have got 2750 amino acids. Okay. What is the weight of the protein? The reverse. So what will be the answer? Calculate.
Zero 250 0.
एक सेकेंड ना. Okay. हाँ, yes Ranjan.
302500.
302500. Huh? And just let me check. Okay. Anyone else?
Same answer I got.
It is wrong.
Huh?
It is wrong. Again, missing one point. See, this is what happens even if you have calculator. Think. I'll give only 1 minute. Ranjan, सोचो क्या मिस हो रहा है? How peptide bond is formed?
Between two amino acids, ma'am.
Exactly. What is that process? What is that chemical process called as? Are you guys even listening when I'm teaching or just listening to the video afterwards? Huh? Sandhya, what is the chemical process that involves in the peptide bond formation?
It involves NH2 group of first amino acid and COH group of this second amino acid, ma'am.
Correct. It is called dash process. What is that dash?
Peptidization.
Arey Baba, no, it is called condensation. ठीक है? Huh.
Yes.
Just now I mentioned it is called condensation. It is a chemical process. So where what is happening? So what happens when the amine group attacks on the OH group? So when this condensation is happening, what exactly is happening? It means what? OH plus H, H2O is removed, correct?
Remove.
Yes,
ma'am.
so with each peptide bond formation, one molecule of water is removed. So how many peptides are present in 2750 peptide protein?
2749.
Very good. So how many water molecules has been removed?
2749.
Correct. What is the molecular weight of water?
18.
Very good. Now calculate. So we don't have that much value in the protein weight. Getting the point? So 2750 amino acids are there. 2749 peptide bonds are formed. It means those many numbers of water molecule has been removed. So that amount, that is 2749 into 18 weight of the protein has been removed from the protein total weight. Correct? Now tell me what is the answer. So you have to subtract the value of water. So that will be around 2749 into 18. That is 49482. So something value of 253018 will come. So this can be written as 253 kilodalton approx. Is it clear?
Yes, ma'am.
Huh? So that small thing is you were missing. Now let's do another question. This time give me the correct answer. Okay. Okay. So you have got 98 amino acids. Okay. Now calculate the weight of the protein. So I have given the small number. So make it fast.
$17.81.
One second. Huh. So what is the weight?
97 into 18 by 98.
By 98.
903.
Amino acids.
Okay, just check with me, okay, whether you have done the same or not. So total weight of the protein will be 98 into 110 minus 97 into 18. Okay. So this is how you have to done. So one amino acid 110. So 98 amino acids weight will be 98 into 110, which is 98 into 110 is 1070, 10780 minus number of water molecules is 97. So 97 into 18, you get 1746. So on subtraction, you get the answer as, okay. So answer is 9034. So approximately 9 kilodalton. Okay. Clear? Anyone is finding difficulty in understanding? Ranjan, clear hai?
एक बार और मैम.
हाँ, ठीक है. So, see, you yourself can take any number value, okay? So let us take another value. Let's take 50 amino acids are there. Why 50? We don't have 50 amino acids. 500 amino acids are there. हमें पता है 1 amino acid is equal to 110 Dalton. So how many will be 500? So the total weight will be 500 into 110. Correct? So let's calculate how much is 500 into 110. That is 5500. But we need to remove the weight of water. So 499 will be the number of peptides, correct? Formed between the 500 amino acids. So 499 peptide means 499 molecules of water will be removed. 18 is the molecular weight of water. So that is 499. Huh?
By doing multiplication, 481 will come in.
So then you will get the answer something like this, and finally some answer you will have minus. Okay, something around 46 you will have. 18. That is Dalton. And every time you express the value of protein in kilodalton. So 46 kilodalton. Approximate. So this is with the help of calculator. You have to learn quick calculations without calculator. Because in the exams it won't be allowed. Now it's clear. Don't worry, I'll give you so many questions today. So today's
Okay,
assignment
ma'am.
I'm going to give you so that you can practice. Okay. And this is what comes from this chapter. They are not going to ask you the given structure is there, so you have to calculate this bond or that bond. No, this is the important questions, and till you do your PhD, this question will come. You won't believe for my case also. So in my case, I had 9 kilodalton protein. Okay, so 9 kilodalton protein means approximately around 89 to 91 amino acids will be there. But I have to be sure like how much it is. So there are certain techniques called MALDI, MS. I don't know whether you have heard or not, but we make use of these MALDI, ESAI to get the actual number of amino acids. Okay. So from this chapter of amino acids proteins, we are going to learn all these things. Eventually, if you go to research field, you have to carry out for your own protein. Okay. Yes. Now there are certain proteins which have got certain prosthetic group. They are called conjugated proteins. So this we already know. It's just that they're associated with certain chemical components. And that non-amino part of the amino acid is called its prosthetic group. For example, if you call it as lipoprotein, then lipids are the prosthetic group. For example, lipoprotein of blood. Glycoproteins, carbohydrates. Phosphoproteins, it means phosphate group is present. For example, the casein, it has got phosphorus group. Hemoprotein has got heme. Flavoproteins have got flavin. Metalloproteins, it means already the metal ions are present. For example, iron in ferritin, zinc in alcohol dehydrogenase. Calcium is calmodulin. So calmodulin already we have discussed. So how many calcium ions can bind in calmodulin? You have to remember is 4. Okay.
4.
Yes. Molybdenum dinitrogenase. Now where do we study dinitrogenase? In the nitrogen cycle.
Nitrogen fix.
Yeah, nitrogen fixing. Yes, very good. And copper in plastocyanin. So today's assignment. Now, whatever assignment I'm giving, in future it is coming. And if you don't remember that time, it is not going to help you then. Okay. Making you understand, I can do, but whatever is there, you have to remember. Okay. Now, what you have to remember is milk, hemoglobin, succinate dehydrogenase, all these. From here to here, you have to remember what is their prosthetic group. Okay. Now, ferritin is there, but how many ion molecules? Alcohol dehydrogenase is there, but how many zincs are there? Calmodulin already said 4 is there. Dinitrogen, how many molybdenum ions are there? Plastocyanin, how many copper ions are there? Now, this is all important. Just make a small chart. I think I can give one assignment to some of you. So, already Akshita has done her assignment, right? Yes. So now I'll pick the name Ramya. Ramya, will you do the assignment?
Okay, ma'am.
Okay. So how many days do you want?
By tomorrow evening.
Yes. Done. Okay. So just make it a proper way so that I can discuss in the presentation because it's important. Okay. Okay, guys. Yes. So let's move on to the next. Okay, so moving on to the next. Now the levels of protein structure. So this I have mentioned once that there are 4 levels in the protein structure. First one is the primary structure, that is your polypeptide chain. So after translation, what you get is the polypeptide chain. Then when it enters the ER, okay, so either the protein has been synthesized on the ribosome itself, correct? Either in the free form or in the bound form of ribosomes. So in both the cases, whenever the polypeptide chain has been prepared, it goes into the ER, then ER to Golgi, correct? Now, these 2 organelles are responsible for your protein modifications. That is proper folding of your protein. So first is the primary structure, that is only the amino acid residues are there. Then alpha helices or beta sheets. Okay. Now, alpha helix and beta sheets are formed because of the hydrogen bonds. So each one of these levels we are going to discuss in detail. Do not worry about that. So alpha helix and beta sheets are formed because of hydrogen bonding. Okay. Then once the secondary structure is formed, then it goes to the tertiary structure. So that is your entire polypeptide chain has been, assembled. So many proteins do function at the tertiary structure itself, and there are many proteins which go still quaternary structure. For example, myoglobin and hemoglobin is there. Okay, so both of them have got the globin as conserved domain. Both are responsible for carrying oxygen. Correct? Now, what is the difference among these two? That myoglobin is your tertiary structure, whereas your hemoglobin is your quaternary structure. So it has got only one polypeptide chain, whereas hemoglobin has got 4 subunits. Clear? So this example will come in the coming slides. So we'll study slowly. So is it clear this much? So once the polypeptide chain has been made, it is only the amino acid sequence. Okay. Then depending on the hydrogen bonding, it either gets converted to or folded to alpha helices and beta sheets. Then again, further folding, the ionic interactions, the hydrophobic interactions, right? All these interactions together make a one complete closed structure, that is your tertiary structure, example myoglobin. Then you have got the quaternary structure, for example as hemoglobin. So where all these tertiary structures like this, 4 units will come together and it will form one hemoglobin. So these are the 4 different levels in the protein structure. Now, whenever we talk about protein structure, you need to have a proteins in their individual form. It means what? It has to be purified form. For example, nowadays everyone is talking about corona, right? And in the case of corona case also, few researchers are focusing on the DNA part or the RNA part. Then few are focusing on the protein part. Okay. So we know that the structure of corona is something of like this sort. So whatever on the top you see is the protein receptor. Now, if you want to study like how the coronavirus is entering into the host cell, the simple thing is that the protein receptor which are present on the top of the coronavirus is interacting with the receptor present on the host. So when this interaction is very strong, what happens? Endocytosis happens. Okay, and the virus enters the host, where the virus will take the control over the nucleus of the host cell and finally infects the cell. And then it keeps on dividing and causes the host cell to lose its own defense mechanism, and hence the infection spreads. Okay, so this is the basic mechanism by which any virus infects the host cell. Now, if I as a researcher would like to study about a protein receptor, what I'm gonna do? Okay, so what I will take, one traditional method is to grow so many of virus, okay, kill them and then purify the protein. But nowadays what we do is, this is lengthy procedure and risky also, because I have to deal with the coronavirus directly. So instead of this, we have got cloning. So there comes your MolBio. So if you know which protein you want to study, get the sequence, insert into the vector. Now this vector you put into a bacteria. Now you can cultivate the bacteria which is going to produce this protein in large amount. Then you purify the protein. Okay. Now let's say that I want to purify a plant protein or animal protein. It can be bacterial also. Okay. So what is the first step in the protein purification? It is your cell lysis. This is your first step. So let it be plant, animal or bacteria or even fungus, whatever that is your interest. Now, what you have to do is lyse the cell. Now tell me, among plant, animal, and bacteria, which is easy to lyse? Which one is easy to kill? Yes. So now we are discussing cell lysis, okay? So among plant, animal, and bacteria, which one is easy to lyse?
Animal.
Yes, Ranjan?
Animal.
Yes, very good. It is the animal. Now, who can tell me the answer for this? Like why?
Due to absence of cell wall.
Very good. So we know that in the case of bacteria, the cell wall is there, the peptidoglycan is there, correct? And in the case of plant also, we have got the cell wall. Totally rich in cellulose, correct? Whereas in the case of animal, we don't have any cell wall, only cell membrane is there. So what you have to do is, from your animal tissue culture, plates are there, right? In that you grow your cells. So it's very easy. Just remove the media. Just remove the media. I'll show how it looks also. It is called tea flask. It is called tea flask, okay? So this is the shape. Now, so this is like I kept in the horizontal, actually, sorry, vertical. We keep the flask like this, okay? So you buy like this. When you place it, you keep it like this. Why? Because more the surface area for animal cells to grow. If you keep it like this, what will happen? I have very small area for the animal cells to attach to the surface and grow. So I'll keep it like this, horizontally. So it is like in the rectangle shape also. So if you keep it like horizontally, you will get more surface area to spread the cells. Now you have got the animal cells. What you are going to do? Remove the media, wash it with PBS. What is PBS? So the steps are remove the media, then wash it with PBS. This is universal step, okay? That's why I'm telling you. Whether you're in abroad or India or any good lab or bad lab, this is the first step. Remove the media, wash with PBS. What is PBS? It is phosphate buffer saline. You have to remember, okay? And what is the concentration? It is called 1x. So stop me in between wherever you get the doubt, okay? So now let's talk about how animal cells are lysed. So first remove the media, wash it with PBS. Then next what? Next, either with the, what to say, the tip is there in which you use the media to remove and put the media. You can take it, you can scratch it with that, okay? Or you can take enzymes called trypsin. It will help the animal cell lines to get detached from the tea flask, okay, from the surface. So trypsin is there. Then just shake it, not shake it, just homogenize it. So what will happen? Just give a little mechanical shear. So what kind of mechanical shear you can give? Mix it or centrifuge it. What will happen? Because of the centrifugal force, the cells are under the centrifugal force, right? So because of that, the cell membrane of the animal cells ruptures. Okay. Then what happens? Whatever you get, you centrifuge it. So this is a symbol of centrifuge. So usually what we use is 10,000 RPM for 10 minutes. Okay. So this can vary from lab to lab. So whatever I'm discussing right now is something which I have followed in my lab. What is RPM? Revolutions per minute. Okay. Now, whatever you get in the pellet is, so you have got your appendix like this. Correct. So on the bottom, you get your cell pellet. This is debris. You don't want them. We don't want that. What we want is you get on the top. So that you take, it is called supernatant. Okay, so this supernatant is you are interested, and that is called crude extract preparation. So you have done the cell lysis, you have prepared your crude extract. So crude extract means it is going to have all the proteins which are present in the animal cell, including the membrane proteins, the cytoplasmic proteins, everything. Okay. Now what you have to do is fractionation. Then something is called fractionation. So what is fractionation? See, in the crude extract, you have got all the proteins, membrane, cytoplasmic, small, big, large, all kinds of proteins are there. Okay. Enzymes are also there. Now, there are two methods: ammonium sulfate precipitation or pH precipitation. So let's discuss ammonium sulfate precipitation first. So what happens in ammonium sulfate precipitation is salting out and salting in. Okay. Now, what do you mean by this? See, this is your salt-free solution, and let us say that these are your protein molecules. Let us say that these are your protein molecules in your crude extract. Now, what you have to do is start adding your ammonium sulfate. Okay. So what will happen? Slowly, the ammonium sulfate is going to take the space between the proteins, removing the water. So what will happen then? All these proteins will start coming together. Okay, surrounded by salt. Can you see it over here? So this will make your protein to come together and precipitate. Okay. And then you have got, this is the another method where you make use of the matrix, but this is not required in our kind of problem. Okay, so am I clear over here what is happening in the case of ammonium sulfate precipitation? So it is called salting out procedure. So what is happening? You have got a crude extract. So it will look turbid solution. Slowly you add your ammonium sulfate salt. Okay. Now, how do you make use of this is, how you're going to make use of this is, that is, see, you have suppose 100 ml of your crude extract. Okay. You have added 10% of ammonium sulfate, AS. Now, when you add, it will get dissolved, correct, initially, and centrifuge again 10,000 RPM. What you will get? Supernatant and a pellet. We are interested in pellet. Take this pellet and label as 10% AS. AS stands for ammonium sulfate. Now, take the remaining ml, add 20% of AS. Now, how do you get this thing? 20% means 20 gram in 100 ml. Okay. So, suppose here it is 90 ml. So, calculate accordingly what will come over here. Are you getting the signs over here? Getting the maths over here? So, you started with 10 gram in 100 ml. So, you did the precipitation, you got something out. Next, once you remove the pellet over here, you are going to take the remaining, let us say 90 ml. Then the value will you have to calculate. Okay. And then again centrifuge 10,000 RPM for 10 minutes. Again you will get some supernatant as well as certain pellet. So I'll explain, one second. Okay, I'll explain again then. Okay. So what is happening over here is, see, till cell lysis and crude extract preparation, it is clear, right? So let us say that you got 100 ml of your crude extract preparation. Now, I have said that it is 10% of ammonium sulfate in the first step you have to add. Fractionation means what? You have to make it into small fractions, right? Small portions you have to make it. So first one is 10% ammonium sulfate. It means 10 gram in 100 ml of the solution. So what is 100 ml solution over here? Crude extract preparation. What is 10 gram? The ammonium sulfate. Is this much clear? So you're going to take 10 gram of ammonium sulfate and dissolve in 100 ml. Okay. Now, what is dissolving? How you add sugar and salt in your nimbu pani? Same way, just dissolve it. Now, once you don't see any ammonium sulfate over there, then you have to take the sample and put into centrifuge. Okay. So in the centrifuge, you are going to get two portions called supernatant and pellet. So since here, because of ammonium sulfate, the proteins are getting precipitated out. So it means it is going to come into the pellet. Okay. So this is your ammonium sulfate pellet in which we are interested. Take the supernatant back. Add 20% of ammonium sulfate to this. Okay. So let us say the value was 90 ml because you have removed something. So from 100 ml, it will get reduced, correct? The volume will reduce. So you can do the cross multiplication, that is x by 90. We don't know how much gram of ammonium sulfate we have to put, and then you can do the calculation like this. Or there are software now available in the internet also. So some value you will get for x. Add that much amount of ammonium sulfate. Okay, dissolve it. Again centrifuge. Again separate supernatant and pellet. So this is your first sample known as 10% AS. Second sample known as 20% AS. Okay, so you keep on doing this till you get 90% AS. Okay, towards the end you will have supernatant and 90% AS precipitate along with 80, 70, 60, 50, 40 till 10%. So these many pellets and one supernatant. Now it is clear?
Yes, ma'am.
Little bit. So you have got these many samples. Okay. Then what you do is called dialysis. It is called dialysis. Now, in this case of dialysis also, this is your precipitate inside. So there is a membrane. Okay. So it comes like this in this shape. The membrane comes like this. It is hollow. Okay. So from here it is open and from here it is open. So what you do? Tie it at the one end, just like this. Okay. Now to the other end, you can add your sample. Now what is the sample you're going to add? All the precipitate from 90% to 10%. Not in the same. So 90, 80, 70, 60, 50, 40, 30, 21. So you are going to have all the 9 samples. So all these 9 precipitate into 9 different dialysis bag. It is called dialysis. Okay. And then one we had supernatant. In that also little bit amount of ammonium sulfate will be present. So +1. So it means 10 dialysis bag you will have. So keep all these dialysis bag. Okay. Now against what you're going to do the dialysis? Dialysis, remember what is dialysis? So inside the bag, that is inside the precipitation, you got the high amount of ammonium sulfate. I want to remove that ammonium sulfate now. So I'm going to do the dialysis against the buffer. Some buffer I'm going to use. You can decide the buffer later also. So usually people use PBS only. Okay. So 1x, or it is also called 10 millimolar PBS. So it's very light concentration. So what will happen? High concentration of solute of that is ammonium sulfate inside the bag and high concentration of water outside. So how is the movement? high concentration of ammonium sulfate will move from inside the bag to outside the bag, or in other words, high water concentration from outside will move inside the bag. Okay, so if you see the figure also, what happens? See, all these molecules of ammonium sulfate has gone out. Okay, and the size of the dialysis bag increases. Of course, right? Water will go inside. So what is happening? Now my sample is free of ammonium sulfate and only interested protein is present. Is it clear? Some. See, I'll repeat once again. This much is clear, right? That you have got 90% to 10% your ammonium sulfate sample is there. Okay. Plus you have got the supernatant. So total 10 samples are there. Take these 10 samples into the dialysis bag. So there is a membrane. In this form it comes. Okay. It's like a long sheet. In a circle it comes. You have to just cut the one end. And one end you have to tie it with rubber band or some clip. To the other end, suppose I have tied from this side. So from this end, I can add this sample, 90% protein precipitate is there. So I'll add into this. Then again, I'll tie it from here. This I'll put into a beaker containing the buffer. Okay. Now, this will be kept on the stirrer. So what will happen? It will just move like this because I need water to get, everywhere. Now, your dialysis bag has got your protein of your interest plus ammonium sulfate, salt. I don't want salt. Okay. So what I'm going to do is, after tying, I'm put into the beaker containing the buffer. Now, what is dialysis? Movement of water from high to low, or in words of solute concentration also, high to low. Now, solute is inside the bag high and outside it is less with respect to ammonium sulfate. Okay. So what will happen once I put into the water or the buffer? Ammonium sulfate from inside of the bag will go outside of the bag. Now the question comes, what about proteins? Why can't protein move outside? Okay, that question should come. Now proteins does not cross the membrane because of the pore size. So the pore size of the membrane is only to allow small molecules like ammonium sulfate salt and water. And proteins are very huge in front of water molecule structure. Okay, and that's why none of your protein will go out of the membrane. So what is happening? After some time, all the ammonium sulfate which was present inside the dialysis bag will go out, and of course in return the water goes inside. Water means buffer. So only your proteins along with your water molecules are present. Okay. Now it is clear.
Yes, ma'am.
Little bit, huh?
Yes, ma'am.
Just go through once. Okay. Listen to the video also once again. I'll put it today itself. And this is the last step. Okay. This slide doesn't mean that this is over. This is whatever till now I have talked about protein purification is only the overview. From tomorrow onwards, we are going in detail. Okay. We are going to dip into the ocean now. This is just the surface. Now, in column chromatography, what we do is the final step for your protein purification. Okay. So here, this is called reservoir in which you add your buffer, against which you have done the dialysis. So once a protein in one particular buffer, you cannot change the buffer. Okay. If you change the buffer, the protein will misfold and it is of no use. Please remember, these are kind of interview questions also. So remember, it is the same buffer which we have used for dialysis. Then these are the columns. Okay. And what you can see is the knob is there to open and close. Now what you do is there is a stationary phase. A solid matrix is present by which the protein is going to pass through. Okay. Now, depending on the protein's property, whether it is hydrophobic, hydrophilic, ionic, whether it is the protein is positively charged or negatively charged, or it is having a protein ligand binding affinity is there. So depending on that, we have got different column chromatography. Okay, so let's just discuss one. So whatever it is, each kind of column chromatography is going to have the stationary phase. Stationary phase means it's a solid matrix to which your protein is going to pass through. So if it is affinity, it is going to bind. Okay. If it is ionic, it is going to have the ionic interactions. Hydrophobic also, hydrophobic interactions will be there. So depending on the property of the protein, you decide your column chromatography. Okay. Then we have got the mobile phase. So on the column, so suppose this is your column and it is having the, suppose this is having the matrix. Now what you do is, so your protein will be there. After dialysis, you got some protein, right? That protein you load on the top of it. So slowly with the help of the pipette, 1 ml, 2 ml, 10 ml, you can add on the top of the column chromatography. So let us say that I have added 10 ml of my protein solution after dialysis. Okay. Now, after dialysis, what will happen? Slowly, these protein molecules will go into the pore size of this matrix. Depending on its property, what will happen? It is going to get separated. Now, the size of the column is big. So, all the protein will get absorbed over here. Now, I need to remove this protein. So, what will I do? I'm going to add the buffer. Okay. So, the same buffer used for dialysis, I'm going to add. What will happen? The protein will start moving downwards and I'm going to collect into the test tubes. I'll check the presence of the proteins in these test tubes either using spectrophotometer or SGS page. Just understand the overview because now from tomorrow onwards we'll do in detail. And SGS page. Yes. Clear? Is it clear till here?
Yes, ma'am.
Yes, ma'am.
Okay. So thank you for listening. Time is also over.